Glycolysis is the oxidation of glucose made by two combined 3GP molecules to pyruvate through a series of steps. The initial steps in glycolysis are the addition of two phosphate atoms to the glucose at the expense of two molecules of ATP.
The result is 6-carbon sugar diphosphate and 2 low energy of ADP. This 6-carbon sugar diphosphate will split into two 3-carbon molecules.
Each of them is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD plus to form NADH and ADP to form ATP.
Begin the Krebs cycle. Pyruvate is converted to acetyl-CoA. In the process, carbon dioxide is released and one molecule of NADH is formed. The 2-carbon acetyl-CoA is transferred to a 4-carbon molecule.
Producing a 6-carbon compound, the CoA carrier molecule is released during the process. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD to form NADH.
The process repeats, but in addition, a molecule of ATP is produced. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2.
These reactions regenerate, and the 4-carbon molecule that initially reacts with acetyl-CoA.
Oxidative phosphorylation, or chemiosmosis, in the mitochondria is the process by which most ATP is produced. Similar to the process in photosynthesis, hydrogen molecules move down the concentration gradient to create ATP from ADP. NADH and FADH2 in this process donate electrons to the protein blobs shown here. This stage consists purely of electron transports into the outer membrane of the mitochondria. At one point, oxygen will receive some energy and bond with some hydrogens to create water. Just a quick caveat is that when FADH2 is used, it will only release a smaller amount of energy since it is further down the chain. Once the concentration in the outer membrane has reached a certain level, they will begin to go back to the membrane through the ATP synthase, combining ADP and a P to create ATP within the mitochondria.